This is a companion video to explain simple genetic engineering for your GMO project in Science 9. Making a genetic modification requires us to be very specific. How specific? We need to zoom in past the cell, nucleus, chromosomes, and long strands of DNA to the genes. Genes are segments of DNA that code for specific products. We want to make our modifications so that we have a single change with a single benefit. No matter what you want to do, the first step will be to identify the sequence and location of the gene you wish to modify. This can be done using gene mapping, but preferably we'd like to use DNA sequencing, as this allows us to target the actual base sequence needed. Now that you've identified your gene, you need to decide which type of modification to make. In all likelihood, you'll either add a new gene, alter an existing gene by either increasing production, decreasing production, or changing the structure or function. Or you might consider deleting a gene, but this is seriously cautioned against. Let's look more closely at the concept of adding a new gene into an organism. So in orange we have the gene that we want, and in purple we have the DNA of our GMO. To get the gene that we want, we need to use scissors of a sort, which is really a restriction enzyme, or a type of protein that will cut DNA. The restriction enzyme recognizes specific DNA sequences, and it cuts it in such a way as to leave a single-stranded overhang. This becomes important when we want to insert the gene into our GMO DNA later. Once we have our gene isolated and purified, we want to insert it into our GMO, our genetically modified organism. And so we're going to cut the GMO DNA with the same restriction enzyme that we use to cut the gene. It's important to cut our GMO DNA with the same enzyme that we used to cut out our gene, because this means it will leave the same sequence of DNA overhanging. Because we've cut the GMO DNA with the same enzyme we used to cut out the gene, the overlapping DNA ends will be complementary. This means they'll overlap and they'll be willing to join together. We'll need to add another enzyme called a ligase to make sure the strands stick. Because this is such a common technique and important for you to understand, let's look at it again using a double-stranded model to better visualize the DNA strands. Now let's look more closely at how we might alter an existing gene by increasing or decreasing the production. If the orange is the identified gene in our organism, this is the upstream region. Upstream of the gene, we often have two sequences, the promoter and the enhancer. Both sequences play a role in expressing the gene. By modifying or replacing either of these sequences, we can change the expression of the gene. Sometimes we just want a lot of the gene product. We can take advantage of something called a plasmid, which is extra DNA in the cell and replicates independently of the chromosome. So this would be an example of what a plasmid looks like, a circular piece of DNA. Once we've isolated our desired gene, we can cut the plasmid with a restriction enzyme and insert that gene into the plasmid using the same technique as the adding a gene section. The product of the gene will now be expressed as often as the plasmid directs it to. In some cases, we want to keep the gene product in the organism, but we want to change something about its structure or function. We do this by using something called homologous recombination. In this example, the green represents our genetically modified organism. And this multicolored piece has our gene that we want to insert into the GMO, and it also is contained in the DNA of our vector. And the green is to help us allow or facilitate the crossover from the vector into the genetically modified organism. When we have homologous recombination, what happens is the similar sequences in the genetically modified organism end up crossing over with the sequences in our vector. So this would be one half of the crossover, and to complete the crossover, you'll see that what we end up with is our genetically modified organism with the purple gene, and then our vector will have the original DNA from our genetically modified organism. The result here is that we get the gene that we want, or the modified gene that we want, into our GMO. It might be tempting to consider deleting or removing a gene, but this needs to be cautioned against because organisms tend to need most of their genes. Remember, we want our modification to be passed on to the organism's offspring. For simple organisms like bacteria, yeast, or algae, less of a concern. But for plants or animals, the change must occur in the zygote stage. This is the only way to guarantee that any changes we make will be carried through to every cell in the offspring. Once you've carried out your genetic engineering, you want to sequence the DNA of your new organism to make sure that you've made the change that you intended to the sequence. If the sequence is correct, you're going to want to use quantitative polymerase chain reaction, or qPCR, to measure how your gene is actually expressed in the organism.